This is Dr. Rupak Kumar, a independent scholar from CDSCO, New Delhi, India, and would like to deliver a talk on design and development of a smart portable device for the rapid estimation of morphine content in glazed fruits and vegetables. Before going to the main theme, here I would like to mention that there is a very common proverb: an apple a day keeps a doctor away, but no one tell you which type of apple do you like to purchase or eat. Either this is the natural one or the glazed one. Most of the consumer prefer to purchase the glazed apple without knowing that there is some adverse health effect. You will be surprised to know that this glazing agent is a synthetic emulsifier called as morpholine, and that is coming from the anthropogenic source of origin. If you see the word morpholine, the name itself indicates its physical, chemical and biological property. Morpho means morphological, oily means a oil just like mustard, coconut oil, and in means amine. So basically it is a secondary amine, and if you see the chemical structure of morpholine, they have the two major functional group. One is the amine group and second is the ethyl group. Due to the presence of the amine group, this chemical has multiple application in different sector of the industry. However, due to presence of the ether group, this chemical behaves as a inert one. These are some of the industrial application of morphine. They are used in the cigarette, they are used in the rubber industry, they are used in the paper industry, they are used in the fertilizer, they are used in the paper, they are used in the textile industry, they are used in the agriculture industry. So the list is ever growing. The concern of interest is that morphine itself not a carcinogenic, not a teratogen, but when it is discharged into the environment, it undergoes a naturalization process, and due to the naturalization process, one carcinogenic process will be formed. This carcinogenic product is called NMMOR, and nitrosomorpholine, and this process is going on also in the environment and also in the biological system. Here I would like to mention that morpholine is present in the glazed fruits and vegetables, and morpholine is also present in the cigarette. If you see the concentration of morpholine that is present in the fruits and vegetable, and if you see the concentration of morpholine that is present in the cigarette smoke, the amount will be the same, that is 0.3 mg per kg. So meaning that taking a smoke or taking the fruits and vegetable, that is the glazed one, the effect will be the same, meaning that there is a possibility that one carcinogenic product will be formed when we inhale the cigarette and parallelly if you consume the fruits and vegetable. Further, it means that initially if you are taking the fruits and vegetable, that is the glazed one, you become the person, then you become the patient, and due to the development of the cancer, you become the disabled. So that is the choice is your, either you are planning to eat the fruits and vegetable, that is the glazed one or the natural one. Keeping this into the mind, we are looking for the some of the development of the some of the assay that will detect the presence of morphine in the glazed fruits and vegetable, and when we develop this assay, then we can optimize it, validate it, and then based on that particular assay, we are planning to develop a point of detection device that will tell you how much concentration of morphine present in the glazed fruits and vegetable. The concept behind the macroscopic assay is that there is one chemical, that chemical is called 1,2-naphthoquinone 4 sulfonic acid, and when this chemical bind with the morphine, a pink color or orange color will develop, and based on this color intensity we can tell you either the fruits and vegetable have the morphine content or not. This reaction will be carried out in the presence of alkaline pH, so that we can add sodium NaOH. The USP of the assay is that it required only 2 manner reagent, and I need only 2 microliter of each reagent. having the reaction. Based on this macroscopic assay, we did a pilot study in India, mostly in the Hyderabad, South India region, South India region, and we found that tomato has 0.17 ppm concentration of morphine. However, the carrot has 1.83 ppm concentration of morphine, and capsicum has 0.342 ppm concentration of morphine. So, meaning that this assay will be quite challenging and quite sensitive to detect the. Also, this report is already covered in the print media, mentioning that whatever the fruits and vegetable that is available in the market, most of the fruits and vegetable has the glazed one, so that fruits and vegetable will be long lasting, and also it will be reach to the longer distance. Based on this particular essay, the essay has been narrowed down, and further the essay has been validated by conducting a pilot study that I discussed earlier. And based on this particular essay, we are planning to develop a prototype that will tell you what is the concentration of morphine available in the fruits and vegetable. The prototype is under development, and we are facing some of the technical aspect not related to the biology and chemistry, but some of the electronics component that I am not comfortable. And whenever that device is not available in the market, so I can advise you, whatever the fruits and vegetable you purchase, please follow the concept of 3W, meaning that wipe it, wash it, then put into the water for the longer time, and then if the device will come into the picture, then we can detect with the help of this particular device. So meaning that an apple a day does not keep a doctor away, which I discussed in the earlier slide. I am happy to answer any further question, comment and concern. Thank you.